Hello, hybrid psychology folks. Hope everybody had a good weekend. We're hanging out in the office here on Saturday, putting together our video lecture. We have Mr. Skullmug, full of Sweet Donkey coffee if you like coffee. Sweet Donkey is the place to get it. And we have our other mascot who also hangs out in my office. Should have used him to talk about the nervous system, because we could have talked about how the central nervous system follows the spine up to the brain, and the peripheral nervous system goes out to the various extremities. Alright, jumping back into brains, let's do a quick review. Remember our nervous system, the parts of the neurons, the nerve cells that handle the information processing. The glial cells provide nutrition and support. There is your neuron with the cell body, the nucleus. The dendrites look like little pokey teeth. And then the glial cells as they help the neuron. Remember we talked about afferent nerves carry information to the brain and spinal cord. They affect you. Efferent nerves carry information out away from the brain and spinal cord. You effect other things. Remember our video, the afferent nerves. Afferent nerves bring the effect of getting his head smacked into his brain. And of course the guy doing the smacking over here is using his efferent nerves to effect the poor guy who's sleeping. Yes, once again, the afferent nerves carrying information to the spinal cord and the brain. He's doing some processing. And then, of course, the final part of the video, where the poor guy uses his efferent nerves to go after this guy. Alright, the neuron structures, the dendrites, the nucleus, the cell body, the axon, that long arm, the axon ending that looks like little alien fingers, and the myelin sheath. That's that sheath of protein that helps insulate and allows the signal to travel along that axon. Information flow from the dendrites. They collect the electrical signals. They determine its time to actually fire. The cell body integrates the signals, sends them out through the axon, which passes them on to another cell. Remember resting potential? We won't watch the full video here, but just remember resting potential is that state of having a charge within the cell.
Unstimulated, neurons maintain a constant electrical difference or potential across their cell membranes. This potential, called resting potential, is always negative inside the cell and ranges from negative 40 to negative 90 millivolts.
All right, that's enough of that. But remember that the signal always travels this direction. It's kind of interesting to note the axon of that nerve cell can be from less than 1 millimeter long, and yes, that's right, to more than 1 meter in length. You could have a single nerve cell almost 3 feet long, extending all the way along your body, one cell. That's pretty incredible. Of course, there you see how these two interact. It's the synapse, that connection or that gap between the end of the axon, those little alien fingers, and the dendrites. And of course we know that what happens there is not the electrical signal jumping from one to the other, but the electrical signal triggers the synaptic vesicles, causing them to release neurotransmitters into the synaptic cleft, and then that connects to the dendrite and tells the dendrite of the next nerve it's time to fire. And when that happens, you have what looks like this. Of course, there you can see your sodium ions moving in and out along that axon, making that signal travel the length of the nerve to release the neurotransmitters at the end of the axon, which you're about to see. right here. By the way, hopefully we've got the audio problem worked out. I adjusted the settings on my software, and so it will hopefully not be jumbling up and making all kinds of crazy noises this time. All right, there we go. So you see the neurotransmitters transmitting. And the neurotransmitters examples, acetylcholine, GABA, norepinephrine, a neurotransmitter that's going to get you charged up. Dopamine, you hear about dopamine a lot in regards to Parkinson's disease, people who shake. You can sort of, if you've ever seen somebody with Parkinson's, as they try to reach out to grab something, their hand will shake. They actually have trouble controlling their movements. And you can think of that in a way, if the neurotransmitters if there aren't enough neurotransmitters transmitting the signal from cell to cell, then you've got a situation where you can't control the movement. One of the treatments for Parkinson's is to increase the amount of dopamine by giving the medication to folks. Serotonin, endorphins, that's that natural morphine that makes you feel high if you're a runner. Oxytocin, example of neurotransmitters. Zombie squid, we've covered that. We don't need to watch them again. So, here we are, talking about brains. Anybody feel like this? I sometimes feel like this, especially when I'm in my 8 o'clock class. The Homer Simpson brain. This is not accurate, actually. There would be a donut in here if this were the true Homer Simpson brain. The brain is divided into several structures, and you may recall we went over this a little bit last time. These structures are the hindbrain, which include the medulla, the cerebellum, and the pons. The midbrain, which includes the reticular formation, and the forebrain, which includes the cerebral cortex, the limbic system, including the amygdala and the hippocampus, the thalamus, the basal ganglia, and the hypothalamus. So there you can see the divisions, the basic divisions of the brain. The forebrain, the midbrain, and the hindbrain. So let's start with the hindbrain first. Of course, the hindbrain, as you might imagine, if you were to reach back and sort of feel where your spinal column comes up and enters right up into the back of your skull, sort of, right here where the coffee handle goes up into the coffee mug. The hindbrain, you could sort of consider the hindbrain to be sort of the less advanced, the more primitive part of the brain. If you're feeling primitive, you could say, I'm feeling kind of hindbrain today. I may actually be feeling a little hindbrain today myself. The hindbrain, the primary structures of the hindbrain, the cerebellum, which is this sort of large, well, I'll say large, it's not that big, but this roundish structure right here beneath the larger structure of the brain. The cerebellum is basically the part of the brain that deals with your coordination, your ability to put one foot in front of the other, the ability to balance. When you think about standing up and keeping your balance, the fact that you can do that without actually having to think about it. You don't have to concentrate in general on keeping yourself from falling down on the floor. That's thanks to your cerebellum. This allows you to have smooth movements to, the old test, and hopefully you haven't, but if you've ever had to do the DUI test, where they pull you over and they have you close your eyes and take your finger and reach and touch your nose. Oh, missed it. I've had too much coffee. But being able to do that is an example of the cerebellum working correctly, which might lead you to conclude that the effect of alcohol hits your cerebellum. We'll talk about that at another point. But for now, the cerebellum, that allows you to be coordinated. Other parts of the hindbrain, or this lower brain, the brain stem, which is not labeled here, but this is where the spinal cord comes up, and then sort of starts to expand. That's the brain stem. If the brain were a flower, this would be the stem. Good way to remember that. And also in the coming off of the brain stem and in the hind brain, you have the pons, which has nerve fibers that are involved in sleep and arousal. So basically, if you have trouble sleeping, have trouble waking up, the whole sleeping-waking process is involved in the pons, which is right there. And then you have the medulla, which is this sort of smaller swelling, as it were, in the brainstem. And the medulla is what governs breathing and reflexes. So when you think about it, you don't have to make yourself breathe, though it's actually kind of funny if you sit there and think long and hard about it. It gets a little weird, if you ever tried that, thinking and focusing on your breathing. Can you imagine how difficult it would be if you had to think to breathe and actually make yourself take a breath? I mean, if you forgot to breathe, you would pass out. We don't have to worry about that, thanks to the medulla, which allows us to breathe. This also governs reflexes. So if you, for example, the reflex when the doctor taps your knee and it triggers a nerve, those sort of reflexive actions originate in the medulla. Now, in this hindbrain slide, it mentions the reticular formation. The reticular formation, according to our book, is actually part of the midbrain. In the book here, it shows the midbrain as just being this section in the slide here, but in the book, the reticular formation is more part of the midbrain, and you can see it extends up into the midbrain section here. The reticular formation is involved in what are called stereotypical patterns. Now think of what do you do that's a pattern that you don't have to think about, but it's something. It's a pattern of movements. What might most quickly come to mind is walking. When you walk, you don't have to think, okay, one leg goes here, one leg goes there, and then my arms swing. Walking is a pattern, a pattern of movements that just repeat. You don't have to think about them. Once you learn how to walk, you can automatically do it. Another great example is riding a bike. How you say that riding a bike is something that you never forget. It just comes back to you once you get on. That's another stereotypical, meaning repeated over and over again, pattern of movements and also holding your balance. When you think about it, riding a bike is really a lot of the cerebellum, which is keeping your balance, and the reticular formation, which are those stereotypical movements, to the point that you don't have to think about riding a bike. You just do it. So that is our midbrain, and you've got another example of the reticular formation, how it is connected to a whole bunch of other sections of the brain and helps to regulate and transmit the input from all of these sensory nerves, these afferent nerves, into the rest of the brain. And then we have the forebrain. The forebrain, sort of the opposite of the hindbrain and the midbrain, which sort of just function without you even being aware of what they're doing. The forebrain is the thinking part of the brain. The thing, essentially, the part of the brain that you're aware of, that makes you, your awareness of what's happening now. The fact that you're thinking about what you're going to do tomorrow, trying to memorize questions for a test that you're going to have this week, trying to figure out how to solve a problem. All those things are involved in the forebrain and what goes on in this part of the brain. If you would look at the brains of various animals, and I think actually in page 62 of the textbook, you see a comparison between a rat cat, a chimpanzee, and a human. You can see the size of the forebrain, indicating much more activity and more complexity going on there. Part of the forebrain is the limbic system, which is described in your book as a loosely connected network of structures which are under the cerebral cortex. Now, here's your cerebral cortex, the big wrinkly part of the brain. And these areas are involved in memory and emotion. And so again, sort of making us who we are, because our memories, our emotions are all part of this whole concept of you, of this concept of me, of who I am. And those structures include the thalamus, hypothalamus, amygdala, hippocampus. Here you see the cerebellum, part of the hindbrain, right down here. So your hypothalamus, that's the section of the limbic system that involves drinking, eating, sex, emotion, stress, reward, basically all of those drives. These are actually things that drive you to act and to do things, to get up in the morning, to feed yourself. You remember the video that we showed of from the movie Firefly, where they had tried to create the perfectly calm and non-violent society, and they gave them the drug so that they wouldn't feel any aggression, and they stopped doing everything. They stopped eating, drinking. They just sat down and died. Well, whatever that drug was, it likely acted on the hypothalamus, which is where those drives come from. The thalamus, which is this section right here, that's the part of the brain that sorts and relays information. You can think of it as sort of a computer server, transmitting information in all directions to different parts. The basal ganglia, which is this section right here, that controls and coordinates your voluntary movements. Now, while you're riding the bike, your cerebellum and the reticular formation may be handling your ability to automatically balance, and also your ability to do those stereotypical repetitive movements, pedaling and walking. As you are riding your bike and you are turning left and right, pedaling harder, pedaling slower, that's the basal ganglia controlling those voluntary movements, not the involuntary, because when you think about it, as you're keeping your balance, those are involuntary movements. You're not thinking as you're shifting and making sure you don't fall over. The voluntary movements, the turning left, turning right, speeding up, slowing down, that's your basal ganglia controlling that. And then down here, the amygdala, basic survival, eating, mates, protecting yourself from rivals. And then the hippocampus, that is where we're going to be storing and processing our memories. Now, here is a look of an actual brain, and there you can get a sense of the cerebral cortex, which is right here. Remember, there's the cerebellum, as you see in that picture. That's the actual cerebellum there. But the cerebral cortex is divided into lobes, and you can sort of see these. You can see the wrinkles in the brain. You can see they're deeper in some sections. And the lobes of the brain are divided into the frontal lobe, the parietal lobe right here. It's sort of, if you could sort of imagine, put your head, your hand on top of your head, sort of right back there where most of us have a knot or a dent. That's your parietal lobe. The occipital lobe, very back of the head, and then the temporal lobe, and that's going to be sort of either side, here and there. So you've got your frontal lobe, your parietal lobe, the occipital lobe, and then the temporal lobe. Kind of think of that as being sort of alongside each ear. And within those lobes, you have different functions. And so here, and again, here's your frontal lobe. Within the frontal lobe, you have your prefrontal cortex, your motor association cortex, the motor cortex, the somasensory cortex, the sensory association cortex, the visual cortex, visual association, auditory, auditory, and so you can get a sense of just from the words that these are sections of the brain dealing with vision, sections of the brain dealing with hearing, sections of the brain dealing with moving around. Don't need to know a lot of specifics. The specifics that you're going to need to know for testing purposes will be included in the the test study sheet for test 2. There's another look at an actual physical brain where you can see the cerebral cortex and the cerebellum down there. And if you were to take that brain and slice it through the middle, this is what you would see. And you've heard the term gray matter as it relates to the brain. You would see gray matter here, basically the color, and you would see a white structure there, and then you would also see some empty areas where that are filled with fluid. There you can see the cerebellum, which is here. Looks like that in the cross section. And here is another look at the brain, and just to give you to show you that these basic structures actually have names. The ridges, or if this were a wrinkle, the top rounded part of the wrinkle are called gyri. A single ridge would be a gyrus, so multiple ridges. These are the gyri of the brain. These sort of deep separations are called deep fissures, and you can kind of look and see how this separates parts of the brain. And then the sulci are the grooves. A singular groove would be the sulcus. All these grooves together, the plural, are called the sulci. So you've got the fissures, the sulci, and the gyri, part of your brain. And here again you've got another illustration showing the frontal lobe, the central sulcus, so that's the separation, that's the fissure, well, not the fissure, that's the smaller indentation that separates the frontal lobe from the parietal lobe. Here you've got the occipital lobe back here, the temporal lobe on either side. These are the fissures between them, and then of course there's your cerebellum. Alright, so the lobes of the brain. Let's talk about the 4 lobes: the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. There are our 4 lobes. The frontal lobe of the brain is located deep into that's a misspelling. I'm not sure where that came from. The frontal lobe of the brain, it's under the frontal bone of the skull. I'll have to go back and fix that. I'm not sure where that came from. It plays a role in the following functions. So again, you're talking right here, the frontal lobe of the brain. It plays a role in forming memory. It plays a role in your emotions. It's also involved in your decision-making, reasoning. Maybe is that why when we're trying to make a decision, we hold the front of our head? Kind of makes sense if you're like trying to think. If you're trying to come up with an answer, you do this. You don't do that. And also your personality. This frontal lobe here plays a big role in who you are, and we're going to talk about that shortly. A really interesting case about how the frontal lobe affects your personality. So there you go, a close-up of that frontal lobe. Now here in the frontal lobe also you have the primary motor cortex, motor meaning moving you around. You have something called Broca's area, which is right there. You've got the orbitofrontal cortex, and the olfactory bulb. And as you might imagine, the olfactory bulb, you think smell, that's going to be connected to your nose, the cells in your nose, your sense of smell. All right, the parietal lobe, which is that lobe right sort of arching over the top of your head, deep is located, boy, I'm not sure what happened to this, deep to the parietal bones of the skull. Must have been something wrong with my frontal lobe when I made these. It's located near the parietal bone of the skull. Plays a major role in the following functions. It's the senses and integrating your senses. So if you think about your sense of smell, your sense of taste, your sense of hearing, the parietal lobe involves in integrating those all together so you can understand if you're hearing and seeing the same thing or things that are associated. If somebody, let's say, what would you see? That somebody claps their hands. You both see them clap and you hear the noise. The parietal lobe involves you understanding that act of clapping, the noise and what you see are connected. Spatial awareness. Perceptions of your body in space related to one another. So the parietal lobe is also going to be involved in your ability to sort of close your eyes, extend your arm, and reach out and touch your nose with your finger. The occipital lobe, which is this section back here, the back of your head, is located, here we have that again, deep to the occipital bone of the skull. I don't understand where that's coming from. All right, we'll figure that out later. The primary function of the occipital lobe is processing, integrating, interpreting visual stimuli. It's sort of interesting because your eyes are up here, but the part of the brain that deals with your eyes and what they see, way back here, the occipital lobe. So you might imagine that if you got a shot to the back of the head, you might see stars because this is the area where your visual processing occurs. A close-up of the occipital lobe. In the occipital lobe, you have your primary visual cortex, your visual association area, which is right there. All right, the temporal lobe, which remember is that lobe that sort of comes down around, sort of in the area of where your ears are. Which should not surprise you to know that the temporal lobe plays a function in hearing. Also in the organization and comprehension of language. So basically, the part of your brain that deals with what you hear and being able to organize language. I mean, it's kind of amazing if you think that we can understand what someone else is saying just based on the noises they make from their throat. The temporal lobe also involved in information retrieval, being able to pull things out of your memory. Within the temporal lobe, the primary auditory cortex, responsible for hearing, the primary olfactory cortex, which interprets smells that come from the olfactory bulb, Wernicke's area, which is involved in language comprehension. Notice, located on the left temporal lobe, just one side, there's a disorder called Wernicke's aphasia, which means you can't understand language. If you were to damage Wernicke's area in the left temporal lobe, which would be the left side of your head here, you might have difficulty understanding what somebody's saying, almost like they're speaking a different language. And here you can see in that left temporal lobe area, there's the primary auditory cortex, auditory, hearing, and Wernicke's area involving the language comprehension. There's the olfactory bulb and the olfactory cortex. So this is the part of the brain that's going to be processing what you smell. This is also an interesting slide. Remember we talked about the structure of neurons and how they're made of the dendrites, who then pull the signals that it's time to activate into the cell body, and then the pulse is triggered. It goes to the axon, which then connects to the other dendrite. A lot of different actual types of neurons. Here is a neuron that comes from the cortex, and you can see the cell bodies there. All these are the dendrites. There's the axon. An association cell in the thalamus. It almost looks like it's all dendrites. Here's a motor neuron. This is the neuron or the cell that you would find in your spinal cord. You can see the cell body there, dendrites, and then the axon. That's a skin nerve, a somatosensory cell. There's the cell body, the dendrites up here, the axon way down here. And this is a cell, I won't even try to pronounce it, in the cerebellum. So you can see here are all of the dendrites, the cell body, and then the axon going in this direction. So depending on what part of the brain or the nervous system the cell exists in, the cells are actually made differently in affecting their function. Now, I don't think in this class we talked about the cortical homunculus. The cortical homunculus, if you would think for a second, if you were to take the brain, and as we've shown here, different parts of the brain are focused on different aspects of your life, of different sensing, touching, feeling, smelling, thinking, emotions, walking, feeling. If the parts of your body, the size of those body parts, were related to the size of the brain area devoted to that body part, what would you look like? You'd look something like this, which gives you a sense of how much of your brain is devoted to your hands. And actually, and if you think about it, you think about how much you do with your hands, touching, manipulating, working, typing, everything involved with the hand. That shows you how much of your brain is really focused on the hand. I was surprised by the lips. Did not realize that there is so much brain power, brain area devoted to the lips. The lips are very sensitive, and so anyway, thought that was interesting. Now it's time to talk about Phineas Gage. Phineas Gage is important because it was Phineas Gage who led us to suspect, or at least proved in a sense, that the front of the head, this frontal cortex, is involved in personality. You remember, if you're going to be an ethical researcher and you want to find out whether or not a person's personality is in the frontal cortex of his brain, one way to do it would be to take a whole bunch of people, damage their frontal cortex, and then see if their personality changes. But that would not be terribly ethical. In the case of Phineas Gage, it was done for us. That's, I think, a mask of Phineas Gage after his death, and that is his skull. And if you will notice, he has a large hole in his skull right there. That hole was made by this thing called a tamping iron. Tamping iron was involved in explosives. Phineas Gage worked on the railroad, and he was... they were setting explosives probably required to blow up a piece of rock. What might happen if you had a large piece of rock. They actually do this. They were doing it out on the interstate on 81 a while back as they were expanding it. But what you would do is you would drill a hole, and in the case of Phineas Gage, it was probably somebody actually using a sledgehammer and a big piece of metal to drill a hole in the rock, and then you would put an explosive charge down in that hole, and then sort of like when you shoot a cannon, you've got to take that ramrod and you push the powder deep down into the cannon, and then you push the cannonball in after it. Phineas Gage's job was to take this tamping iron, and after they drilled the hole in the rock, he would take the tamping iron and he would push the explosive charge down into the hole, because you wanted to get the explosive charge as deep in the hole as you could so that they would fracture the rock. And what happened to Phineas Gage is, as he's taking this tamping iron and pounding it down into the hole where the explosives are, the explosion went off. The tamping iron shot back out of the hole and right into Phineas Gage's head. And here you can see a digital recreation of what the tamping iron did. It shot, I don't think it actually stuck in there. I think it shot right through like an arrow. So you can see it went in right below his eye, went up through the front part of his brain and out the top of his skull. And of course, if you look here, you can see it went in here and up here. The bizarre thing is that he never lost consciousness, or at the very least not for very long. Didn't suffer pain, and of course remember, no pain nerves in the brain, and so that's why a surgeon could actually open up your skull and touch parts of your brain. You wouldn't feel any pain. But when a doctor saw him, he was completely normal. Amazing that he didn't get a massive infection, but maybe the explosion sterilized the rod. But went back to work a few days later and seemed completely unaffected by the accident. This is actually similar to what happens in a lobotomy. But going back to that, his personality, however, even though physically he didn't appear to be negatively affected, his personality suddenly and radically changed. His ability to reason, his ability to follow social norms appeared to have been destroyed. He had no more interest in his hobbies. According to the description, he became a nasty, vulgar, irresponsible vagrant. His former employer said he used to be the most efficient and capable foreman prior to his injury, refused to rehire him. And this is apparently similar to the effects of what's called a frontal lobotomy. It used to be that in mental patients, if you were having a problem with aggression, you couldn't be controlled, couldn't relate socially, and what they would actually do as a last resort is they would take a piece of metal and they would actually pound it right into your skull, and they would destroy that frontal lobe or parts of that frontal lobe of your brain. That's why it's called a frontal lobotomy, and it would change your personality. It's not something that happens much at all today, and really it's not needed because of the medications that we now have that do a much better job in changing people's personalities when they have issues. And I think the research would indicate that the lobotomy really didn't do that much to improve people's situations. It may have made them easier to control, but it didn't improve their quality of life one bit. It was mostly just like tying them up and leaving them in a room. So, thought question. I think we've talked about this in class before. And as we've been talking about these parts of your brain that do all these different things that are involved in memory, in what you see, in what you hear, it kind of begs the question, how much of what you perceive in your brain is really what's happening outside your body? Is what you see what's actually there? Have you ever seen something that wasn't there? Have you ever not seen something that is there? The example that I'd like to use is your nose. Do you see your nose? When you think about it, it's right below your eyes and within your field of vision. In fact, if you close one eye, and you can actually look and you can see the side of your nose. You close the other one, you can see that. And if you try, you can see it. But generally, when you're looking around, interacting with the world, as you're looking at the screen right now, do you see your nose? You don't. If you choose to focus on it, you will. But generally, your brain subtracts your nose from your field of vision. So your brain can control what you see. Couple of examples. I want you to look at your screen as I change the slide. and stare at the plus sign. And you will, as you stare at it, you'll be seeing a green, and just keep your eyes focused on the plus. And around your vision there'll be a circle. Now you see that green dot that's circling around? And eventually, if you keep your eyes focused on the plus sign, the green dot will start to, almost looks like a Pac-Man, like it's eating up the purple dots. Stay focused on the plus sign, don't move your eyes. The purple dots will start to disappear, and the green dot will be all you see. What's interesting about that is the green dot doesn't exist. There is no green dot. If you focus back on the image, you can see that all that's really happening is the empty space is rotating. There's no green dot, there's just empty space. But if you stare at this image, eventually all you'll see is the green. So what you are perceiving in your head is not what's actually going on in reality. All right, here's another one. Now, this again requires you to focus very closely on the image and just don't blink. Just keep your eyes focused on the image. Here we go. Now, at some point, as you focus on this image, you will see a giraffe. There is a giraffe in this image. Keep focusing. Eyes open, don't blink. Okay. That was a joke, but yeah, I figured we've been doing this long enough. We needed a little comic relief. Now this is real. Look at the circle. Is that round? Doesn't look like it's round, is it? Well, I will tell you that if you were to get right up close and stare specifically, in fact, if you can maybe take your hands and block out the rest of this, you will see that this is a perfect circle. But combined with these other images, it appears through my eyes, or actually in my brain, to be sort of lopsided, almost egg-shaped, when it's not. This is the exercise of your visual perception and your decision-making skills. The visual cortex in the occipital lobe, remember that's the back of the head, is processing the input from your eyes, and then it's looking at these shapes and colors, and this is messing with your visual cortex, and so it is actually seeing a shape here that is not accurate. That's showing you sort of a lopsided shape, or at least the perception of one, when that shape is in fact completely round. All right, here's another exercise. I'm going to click next and count the number of F's in the following sentence as quickly as you can. All right, how many F's did you get? Almost everyone guesses 3. Apparently the brain does not correctly see or process the F in the word of. And the reason is we think of F as making a F sound like fox, but in of it makes a V sound. So in looking and counting the F's, your brain is not perceiving this F as an F because it makes the V sound. All right, here's another one. This is your occipital lobe working. Look at A, B, C, and D. Which one of these, if you were to take and fold these into a box, would make this box? You maybe even feel the back of your head heating up, because this is making your occipital lobe visualize moving something around in space. This is actually hard for me. I don't do this well. But eventually, as you stare at this box and think about how these shapes and the circle are related, you will come to figure out that if you would fold D into a square, you would get this. However, you can also fold C into a square and get the same thing. That is your occipital lobe processing. Here's another exercise. Count the number of times the number 6 appears, and then count the number of 3s and 7s and try to add the total number of both as you see them. So count the 6s, and then count 3s and 7s. And I'm not going to ask you how many you got, but just know that this exercise is focused on your frontal lobe and your parietal lobe, the front of the brain and the parietal on either side. All right, frontal lobe, working memory because you're trying to remember as you're counting how many you've counted, and attention, and the parietal lobe, visual, seeing what you're seeing here. All right. Interesting to know that if you are right-handed, your language mostly stored in your left temporal lobe. If you're left-handed, your language is probably stored in a bit of both. Words in the brain, it is interesting to note, that are not stored randomly, all over your head. Words seem to be stored in an organized way, so that words that are heard together, salt and pepper, nurse, doctor, seem to be connected. Here's a brain exercise. This stimulates those connections in your brain. So in the left column, you'll have a pair of words. Your goal is to find a third word that is connected with or associated with both of those words. See if you can get the associated word before I say it. Lock, piano. That, of course, is key. Ship, card. Get that? And that one's a little more difficult. Board. You board a ship. Cardboard. How about tree and car? Tree, car, they both have a trunk. Actually, boarding a ship and cardboard, there's another one that makes more sense. Ship has a deck, and you have a deck of cards. School and eye. Well, how are they? A pupil, exactly. What about pillow and court? Pillowcase, court case. How about river and money? Yep, both have a bank. Bed and paper. Both have sheets. A sheet of paper, the bed has a sheet. Army and water. Tanks. Egyptian and mother. Oh, forgot tennis and noise. Tennis and noise. Racket. There we go. Egyptian, mother, mummy, of course, and then smoker and plumber both have a pipe. That's your temporal lobe, pulling those words and making connections. All right, Stroop test. Now this is fun. See how well you can do this. What you need to do is start from top left, and as you would be reading these words on a page, go through these words one row at a time, but don't read the words, say the color instead of the words you read. All right, I'm going to go ahead and start. Having trouble? Let's see how far I can get. I did this earlier and didn't do very well. So I've got red, white, green, brown, green, red, brown, white, red, white, green, brown, white, green, brown, red, brown, red, green, white, green, white. You see, I'm slowing down. You see how difficult that is? You can really get a sense of how your brain is struggling between processing the reading part of what you're seeing and the color part of what you're seeing. So again, you sort of almost feel the different parts of your brain fighting each other. All right, it's been a long lecture, so we will wrap this one up. And here is the question that I want you to post an answer to on this discussion board. This is a bus. Which way is this bus traveling? Is the bus traveling to the right, or is the bus traveling to the left? There is an answer. So click reply in the discussion thread and tell me which way this bus is traveling and explain why. And then on Friday, as we meet in class, we'll be talking about the endocrine system, the plasticity of the brain, what that means, do you have a plastic brain, and then diagnostic tools that are used to look at the brain. And hopefully, if you are reading this or watching this on Monday or later, your grades should have already been posted from your last test. Anyway, thank you for your attention. Sorry for the occasional mistake. Hopefully the audio worked better, and I will see you in class on Friday.